The idea is to put a disc that has been soaked in a plant extract on a petri dish containing bacteria culture and see if it will stop bacterial growth around it. Before I start, let me mention that if you'd like to read our books on the internet, find us at booksy.com. You can write Msamarumema Herbal Cure and you will see all our books. Many people are reading our books. By the time of posting this video, 5,819 people had read our books. For example, 713 people had read about origins of cancer, our book about origins of cancer. 661 had read about costly beauty. We have got so many books right there. Book range is wide, so you can read all topics but centered on healthcare. Secondly, we also have other Facebook pages apart from Msamarumema Herbal Cure Journal. You will find Cancer Kenya, Blood Pressure Kenya, Diabetes Kenya, Arthritis Kenya, Msamarumema Green Foundation, Asthma and Allergies Kenya, and we also have a group page, True Herbal Kenya. So, how do we do drug sensitivity test on our herbal medicine? As a well-trained herbal doctor and medical microbiologist who did lots of pharmacognosy, I can show you how we determine what drug to use in what infections. This is done through drug sensitivity tests. This line of approach, I did it at Kenya Medical Research Institute, KEMRI, Center of Microbiology Research, CMR. First, a big shout to Said, who guided me through at KEMRI. First, before you start drug sensitivity test, you must know your drug source well. In pharmacognosy, you must know your plants and the targeted chemicals in those plants. Certain plants have antibacterial properties, others have antiviral properties, while others have antiprotozoal properties, and still others have antifungal properties. Today I'm going to show you how to test plants that have antibacterial properties. Bacteria cause so many diseases, respiratory diseases, e.g. coughs and pneumonia, as we see in Streptococcus bacteria. Bacteria cause also sexually transmitted infections like syphilis and gonorrhea. They can cause food poisoning diseases like Shigella and Salmonella typhi that cause typhoid. Bacteria also cause brucellosis like Brucella abortus and Brucella melitensis. Bacteria will also cause abscesses, etc. Kenya Medical Research Institute, KEMRI, has all these types of bacteria stored and ready for use in research purposes for people like us to use them for research. So you don't need to go out there looking for patients and collecting the bacteria. They are already there. You must also know the plants that contain specific antibiotics and antibiotic properties that you want. Plants that you use are the ones that you think can treat particular diseases because of their chemical properties. For example, Drosera species, this plant contains the plant chemical plumbagin, which kill bacteria that cause respiratory infection. Melissa officinalis contains the chemical thymol. It is an antibacterial. The plant pumas has antibiotic properties against gonorrhea. It also has the plant chemical ascaridol, which kills the bacteria Staphylococcus aureus and can also kill the bacteria Pseudomonas aeruginosa. There are so many plants that are antibiotic, and to keep this video short, I can only mention a few plants. Filipendula treats food poisoning. Thymus, Viola tricolor, Humulus, Gentiana, which treats ear infections, etc. So what do you need to do drug sensitivity tests? You will need discs that have been soaked in specific plant extracts of interest. This is done in sterile environment. These discs are stored in vials. Like here we have 6 vials with the discs from different plants of interest. You'll need gloves, you'll need tweezers, sterile cotton swabs, you'll need a marker pen, you'll need a petri dish. This is a plate, a petri dish with a single colony of the bacteria that you want to test. You'll need a blank petri dish with agar media where you will grow the bacteria and do the test on. You will also need a waste container. What do you do then? Put on the gloves, label the petri dishes at the bottom by writing the initials of the plants that you are testing. Take the sterile swab stick and draw a black dot on one side of the stick. This helps you to know which side of the stick is inoculated. Take a single colony of the bacteria, the black dot facing you. Inoculate alone on the blank plate by spreading the bacteria evenly. You can spread the bacteria again in the perpendicular direction. Discard the used swab in a waste container. Divide the inoculated disc into 6 parts. This helps you to know which disc is placed where. Use the tweezers to pick the antibiotic disc and place it in one of the divided sections. Each disc is labeled with an initial of the specific plant chemical. Do the same to each of the other antibiotic discs. Lastly, place the discs upside down in a 37 degrees Celsius incubator for 48 hours. After the bacteria have grown in those 48 hours, you will see a clearing around the antibiotic discs. These are called zones of inhibition. This shows how good the plant extract in killing particular bacteria, therefore its ability to cure a particular disease. You can measure the strength of the drug by measuring the diameter across the zone of inhibition. And measure in millimeters. Then use a reference table to see if it fits the set standards of susceptibility of that bacteria for a particular plant chemical. Please like, subscribe, and share. What might seem trivial to you might actually save a life. See you in the next post.